go.
Today we are going to go through the enzymes lab, demonstration of how the enzyme lab is done and how we are preparing the chemicals, how we are measuring the enzyme activity. So the enzymes are kept as a diluted form, and this is a glucose oxidase enzyme, and the substrate is the glucose, that is the sugar that we consume. It's a 6-carbon sugar. The enzyme glucose oxidase break down this in the presence of oxygen, and then it releases the hydrogen peroxide, and we are trying to measure the activity of this hydrogen peroxide after its glucose oxidase work using the compounds called phenol and 4-amino antipyrine. So that produces a color. The more color there is, the more enzyme activity. Okay? The main objectives of the lab that we are doing today is to study the effect of concentration of the substrate, which is the glucose, on the activity of the enzyme, and then we are going to look at the effect of temperature on the activity of the enzyme, and then the effect of pH on the activity of the enzyme. So these are the main goals of this one. So to start with, I'm going to do the effect of substrate concentrations on the enzyme activity.
2, 3.
So the first part of this experiment, we are going to study the effect of substrate concentration on the enzyme activity. So you want to be always careful when not to mix up the substrate with the enzyme activity, otherwise the reaction will start right away. Okay? So what I'm going to do is to label the cuvettes. I'm going to label the cuvettes based on the concentrations, but you can also label them, with a specific number so that you know which one that you are using for which concentration. So the concentration is measured in milligram per deciliters. So we're going to go from 60 to 30 to 15, 7.5 and so on. So each time when you dilute the concentration, it is going to become half the concentration, but the dilution factor is 2. So to have this, you can use the formula V1 plus V2 over V1 is equal to DF, and then figure out what that V1 is, and then you already know the V2, which is a 1 ml. So in this case, it's a 2-fold dilution. You mix 1 part of the stock with 1 part of the water. But we are doing a serial dilution. In this case, you add the stock only once to the first cuvette, then you add water to each one of the other cuvettes, mix and move, and then discard the rest. Okay, that is what I'm going to show you, how to do that for serial dilution. So this is the stock, and here's this micropipetters. The micropipetter, you can set it up with the dial on the top, and you can change this. Now it reads 1000. That is going to measure 1000 microliters equal to 1 milliliters. So I'm going to use a blue tip for this micropipetter, and the blue tip protects the micropipetter from being contaminated, and that is a right way of using this. Also, whenever you're using the micropipetter, you want to keep it in an angle like this and use your thumb to take the liquid in and to eject the liquid out. Don't ever do this. This is going to contaminate the micropipette. These are simple tips that you want to be careful about. Also, some students do this using their pointer, which is not efficient, so you get tired soon. So here's a micropipetter. Here's the glucose. You want to make sure you're adding the right amount, and I am going to, and also you don't want to leave this like in a jerky motion because it will take air in. So I'm going to do this very gradually, and the liquid 1 milliliter is taken in. So now I put the glucose back on the ice, and now I'm going to add it to this first dilution, and I have 1 milliliter of glucose. So to this, I'm going to now eject this tip, and I can use this button to eject this into a waste beaker. Now I take a fresh tip. So this is something important. You want to change the tip as much as possible, and also wear gloves to avoid contamination of the enzyme or the reaction. Now I'm going to take distilled water. So this is how you do the dilution. 1 part enzyme, 1 part of water. So now the concentration becomes half, the dilution becomes twice or 2x. So now I mix the two together, I discard the tip, I put the micropipetter back on the stand, and that's also avoid any contamination. There are little small closures that you can put on these cuvettes, and these closures will help you, do this dilution by mixing this. And there are other types of a way of mixing this also by pipetting in and out, or if you're using a test tube, you can use a vortex machine to mix this. Now I have the first dilution done. So the next thing I have to do is to move 1 milliliter into the next cuvette. I'm going to add 1 ml water to each one of them now, so that it's easy to mix, move, mix, move, mix, move like that. So I'm going to add 1 ml water to each one of the other cuvettes, then it becomes a lot easier. Okay. So the other thing you may be able to do is, instead of using these closures, you can also use a paraffin. So that's like a waxy type of closure, and that also you can use to do this mix and move instead of this plastic closure. Okay. And I have done all the way up to 5 and then 6, and then I'm going to do the 7. Okay. So now I have added 1 ml water to each one of them, and then I'm going to take a fresh tip, and then I'm going to go to the first tube, which has 2 milliliters. I'm going to take 1 milliliter from this and move it to the next cuvette. Okay. So here, so this one I'm mixing. See, I can do this type of mixing also. It gives an even mix, or you can also close this cuvettes like we did before, and you can also invert this upside down and mix it. There are 2 ways you can do that. And the other method I said is through because this gets very tight, but you can remove that. Now I have to do the next milliliters, and so this is 2 ml. I'm taking 1 ml from this and go to the third one, and same way I am doing this pipetting in and out to mix this. So this has mixed evenly well, and now I take 1 ml from this and then go to the next cuvette. So this is what is referred to as a serial dilution. So you are doing this in a sequential manner, and the concentration at each stage becoming half, and the dilution is 2x. Okay. So we have the 4th concentration done, and I'm doing the 5th concentration and mixing it well. Now I go to the 6th concentration, and this also you have to make sure that you're mixing it well. Then go to the last one, the 7th concentration. So the concentration are becoming half and half, like 60 became 30, became 15, and 7.5 and so on. At the end, you want to take that and then you want to discard that last one into a liquid waste, and we can discard all these things later on, and also the tips. Okay? So now we have all this concentration done. Then only I'm going to add the enzyme to do the reaction.